All right, so talked about copying the DNA, okay? The synthesis part, right, of the whole interface thing. So now let's look at how we talked about these processes here. Now we've synthesized it, we've divided, okay, cells are proliferating. So now let's see what happens after we've proliferated, and then the cell is just hanging around, and it's like chemical gets released into the body, and it's like, I need this thing made. All right. Let's just say there's a foreign invader. Okay, you have allergies, right? That's a foreign invader. Excuse me. So we have this foreign invader that gets in the body. It has different chemicals on its surface, and that alerts the body that something's here. That tells the body that I need to start making things. And the only way we do that is look at the blueprint, right? The blueprint is in the DNA. So the first thing that is going to happen is transcription. The second thing that happens is translation. And then really, I guess the last thing ultimately is the expression of the protein, but I'm not asking about that necessarily. But transcription and translation, you're damn right. So this is the first step in us making something inside the cell to do something. We're making proteins. Transcription. Where does it occur? A good question. A good part of this big thing that you should know. And what is it? When you transcribe something, you are copying it down. Okay. When you are transcribing something, you are writing it down. All right. So all you're doing is copying. And that's what we do here. We make a copy of one of the strands of DNA. That's what we do. Package it up into what we know as messenger RNA, and then ultimately that's going to leave the nucleus through those nuclear pores found in the nuclear membrane. Connecting dots from earlier in the semester, right? All right, so let's look at that. Excuse me. Because it sounds really easy, right? Of course, it sounds really easy. But of course, there's a whole buttload of steps. So let's get into this process. All right, so what is it right there? A segment of DNA serves as a template for the production of an RNA molecule. We are making a copy. To do that, we do the same thing that we basically have been doing. We have to unwind it, we have to unzip it, and we have to expose it. We have to expose it, right? It sounds so dirty. But we have to expose those base pairs so I can get in there and I can make that copy. Remember, C goes with G, A goes with T if it's copying the DNA. But what are we copying this onto? A messenger RNA strand. It's going to take the message out into the cytoplasm. So instead of thymine, we have uracil, right? So A has to go with U. So don't forget that, okay? Don't forget that crap. That is something that is very commonly missed on the exam. It seems so, you're like, duh, dude, I got it, man. But like, you're going to forget it on the exam. And it's easy to get nickeled and dimed to death on these exams, as you've seen so far. So just those tiny little points, don't give them up, because that's the difference between an A and a B, or a B and a C for a lot of you guys. So don't give up those little bitty pieces. When that entire segment of that gene has been copied, transcribed, I'll just say copied above it, okay, because that's what we are doing. We are making a copy. It gets packaged up into messenger RNA. And then they call it like, well, it's not leaving yet. All right. So we call it pre-mRNA transcript. It's the transcribed portion of the gene. It's mRNA. I don't know why they have to make it so difficult and call it mRNA transcript. It's mRNA. It's just not done yet. As with anything in the human body, when you first create the cell, it has to be fine-tuned. Sperm is a great example of this. Eggs are a great example of that, right? We go from spermatogonia, and then we go through multiple different steps, primary, secondary, until we get the spermatid. And even then it has to fine tune itself before it becomes a mature sperm. mRNA is the exact same way. When we first make that copy, it's the copy, but we've copied literally everything in that segment that's associated with that gene. And we're going to find some of that stuff is bogus. Okay. It's just, well, we call it junk DNA. We call it junk DNA because we don't know what the hell it codes for. It doesn't code for anything, but it once did. And that's what they're doing a lot of research now in that whole genome project for every single animal on the planet. So pretty interesting stuff. Again, there are hundreds or potential thousands of genes that are going to be copied at a time there. So it is a long strand of material that's being copied, even though all the videos that I show you, I mean, it's just a small little handful of amino acids that get copied. There's hundreds of them, thousands of them sometimes that have to get copied. So just have that in mind, that this is not just, 5 amino acids being copied. Whoopie-doo, who cares, right? There's a lot of them. There's a lot of them being copied at any given time. Again, that genetic information in that gene is an amino acid sequence, okay? And what are proteins made of? Proteins are made of amino acids. Are made of amino acids, right? Those were the monomers. Okay. We copy one segment of the DNA. That's the sense strand. I don't know if I necessarily asked that specifically as a multiple choice question at all, to be honest. It's just the mRNA is copying one strand. That's it. It is kind of interesting how they visualize it. They visualize it as a sentence. So if you have, let's say, 1,000 amino acids there, 1,000 different genes, a handful of amino acids, it is a sentence. And those U's and A's and G's and C's code for various different words. And we saw that at the very end of Monday's lecture, right? With the genetic code, that universal language, right? All those letters creating words within that sentence. And all those words within that sentence tells the cell what to make. Sometimes it's to make receptors for the cell membrane to mount an immune response. Okay, but it is that sentence that is basically what we are copying down so that the cell can make something in the cytoplasm. All this looks the same as we talked about when we talked about synthesizing DNA. Okay. Instead of the whole DNA ultimately unwinding and being copied, it's only a specific region. Okay. Only the gene to make the protein is being exposed. Okay. Unzip that region, straighten it out, open it up, and copy one of the strands within the DNA helix. RNA polymerase is there. I mean, there is nothing different from what we talked about on Monday. We're just now talking about a specific process that, again, is happening within the nucleus, but now we are only copying one strand. Okay. And the copying of that one strand is what gets me mRNA. And that's what transcription is. We copy a segment of a gene, package it up into that copied is called messenger RNA. Okay. And now after we've copied it, we are going to manipulate it a little bit. Okay. And I mean, there's DNA, there's RNA polymerase, DNA polymerase unzips it there. I mean, nothing is different here. Okay. We saw this the other day. So really there's nothing different. The book gets into initiation, elongation, termination. At this point, I don't really care. I want you to have the appreciation of where this is happening. What is this thing? Okay. What is basically happening? It's the same thing we saw with DNA being doubled up. except now we're only doing one side. We're only copying one side. Only one thing is making sense here, the sense strand. And we make sense of it in the form of a messenger RNA transcript.
During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination.
Right.
During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind.
I mean, if we know the English language, we already know what an initiation step is, right? It's the beginning. What do you think the termination stage is? The end. Okay. The elongation, just the growing read of the mRNA transcript. So if we know what words mean in the English language, you all can do a fabulous job at shucking and jiving and BS in an exam, right? So yeah. So all you have to do is understand what these words mean and expound upon them using the words that we're talking about here, specific with these various different things.
This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. During transcript...
All right. I've just unzipped it, unwound it, unzipped it, exposed it, copied it. Transcription at its most simplistic. But that was referred to as pre-mRNA transcript, right? We have to process it, just like we do with any other cell within the body, okay? It has to be modified and manipulated just a little bit so that it can go out into the world and do stuff. That's what we have to do here, okay? With that pre-mRNA transcript, we have to do a couple things. We have to put a cap on it, and we have to put a poly A tail. Think of it as getting dressed up to go to a fancy ball, okay? You put your top hat on, and you put on your long-tailed jacket. I don't know. That's the parties that I have to go to. That's what I have to wear, okay? A big top hat and a long-tailed party jacket, okay? And that's what we have to do here, all right? That cap on the 5' end, okay, the hat goes on the 5' end. That's why it's there, all right? That's why it's there. It's basically going to tell translation, hi, this is the head of this mRNA molecule. Start here, please. That's what the cap does. For the poly A tail, and it's just a bunch of A's, like 150 to 200 adenines, basically, stuck on the end. That's all it does. It helps to facilitate transport of mRNA out of the nucleus, okay? Excuse me. It also does some, prevention as well. It helps to prevent that 3' end from being degraded by the various different enzymes. But let's just look at the one that I underlined. Just remember that one, and I will be happy. I will be so happy. Okay. So I've unwound, unzipped, exposed, and now copied my mRNA transcript. Initiator, elongation, termination. Once it detaches from that DNA molecule, it gets modified. I put a cap on it, and I have a poly A tail. The cap is what tells the translation process where to start or where to begin the read at the head. You always start reading from the beginning of the sentence, from the head of the sentence, right? Poly A tail just ensures that this thing can get out of the nucleus effectively. That's where we are. Okay. Are we done yet? Of course not. This is biology. There's a bunch of steps involved with it. So of course there's more stuff. There are hundreds to thousands of genes that were copied. And it's not a stream of consciousness that is from the initiator to the terminator end region. All of those letters in there are going to be copied. Some of them are good information. Some of them don't code for anything. That's where we get exons and introns, all right? So I've copied the whole damn thing. I've modified it, put a cap on it, put a poly A tail. It's ready to shuck and move. But hold on. I don't need everything going out into the cytoplasm. The things that get expressed, and by expressed, these are the things that are going to be copied in the cytoplasm, or translated, I guess, in the cytoplasm, are the exons. All right. The introns are found in between the exons. Okay. The introns are basically in the way. They get snipped out. Now, there is a lot of fascinating discussions on what introns actually do. They are still part of, they just don't code for anything. Okay. They're still the A's, the G's, the C's. They're all that nonsense. They just don't necessarily code for anything right now. They may have at one point, and I'm still trying to get that stupid video to work. I'm having a hell of a time. I'm trying to pirate it, but don't tell anybody. What these introns potentially do is they create switches, and sometimes these switches can be turned on or off and create a totally different body plan. Let's look at fish. In some fish, there's this one fish species that there is a freshwater version and there is a saltwater version, and they look exactly the same. They have everything similar. It's just that this one group got cut off when the land cut it off, and that body of water slowly got rid of the salt within it and became more freshwater. And over that time frame, they became isolated and started to change a little bit. And the only thing that they change at is in their body plan, with the presence of these little bitty stickers on the underside of their bellies, for lack of a better word, okay, on the bottom of their body plan. And the reason why they adapted that way is because now that they're kind of blocked off from all of those predators within the ocean, there wasn't a need for these big stickers to come out and protect them. So something happened. There was an adaptation, there was a change, there was a mutation that occurred, and that switch got shut off. And now those little sticker things don't go there, but you still see them on the underside of their body plan. Whereas the ones that are out within the saltwater, exposed to bigger fish, more predators, they still have them. And they've been able to figure out where that specific difference is. It's really fascinating, to be honest. And when we start kind of looking at evolution in the last segment of the class, we'll look at, some of the ingredients to get that just tiny little shift, to get a different little species to kind of emerge there. So it's pretty interesting. So if I put my blinker back on and get back in my lane here, what am I saying? I'm saying that we're still processing this pre-mRNA transcript, okay? I've copied the whole genes that I wanted to. Put a cap, put a poly A tail on. Says where to start, gets it out of the nucleus effectively. But I'm still not done, okay? I need to cut up and trim it up just a little bit to make it presentable for the party. So I have to get rid of those introns. Introns get in the way, blah, 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 blah. Who cares? We need to get them out. So we do that process in the second part of the prep for the party out in the cytoplasm. Depending upon if you are depending upon if you are a eukaryotic organism or a prokaryotic organism, will depend upon really, how complex this process can be. If you are a prokaryotic organism, you have self-splicing capability, okay? But your genome, your nucleus region is pretty basic, right? Binary fission is pretty easy. Copying of the DNA in a prokaryotic organism, right, was super easy. Always starts at one spot, makes a complete copy. Yawn, boring, right? So they can easily do it because they are so basic. For us, we have to have some specific bodies, and some means bodies, okay? BODIES. And these are just cellular bodies that go in there and splice this up, okay, to remove those introns. So this is how those introns are getting removed, by these things called a spliceosome. Splice means to cut. Some means body. So we just have some cellular bodies that go in there and snip out all of these non-coding pieces of genetic material, the introns, okay? So eukaryotic organisms need specific cellular bodies, blah, blah, blah. Prokaryotes, they can just cut it out themselves, okay? They have the capability to just enzymatically just remove them there right away, where we have to make some cellular bodies, splice them all back together. And they splice them all back together by small nuclear RNAs. I don't really care that you remember that, okay? So here's my DNA copy, okay? Here's my strand of DNA that I want to copy, right? I've unwound it, unzipped it, exposed it, copied it. There's my pre-mRNA. Copied everything in that gene region that I wanted to copy. Got some good, got some bad. Poly A tail. Make sure it gets out of the nucleus properly. And my little 5' cap to say, hello, I start here. What else do I got to do? Well, if I'm a eukaryotic organism, I have a cellular body, spliceosome, that goes in there and cuts out the intron. Puts it right back together. And now we are ready to throw a party in the cytoplasm.
The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. The strand
Even though, I mean, even though these introns don't necessarily code for something now, that's why I keep saying now, it may have coded for something at some point during development, or hell, it couldn't have coded for a damn thing. But what is it though? It is the genetic verbiage, right? For a mutation, for a potential different protein, for a potential different change in body plan. Okay. So even though we cut these out and get rid of them, they can do a bunch of potential things, and that's where a lot of research is right now. Yes, it gives us more information for the entire genome. Okay. So let's say there is a misread for whatever reason that isn't caught. I still have all of these letters again, and it may code for something else that can give me something similar or something slightly different that gives me an advantage. And then my DNA is getting, into the next generation because I get to have all the sex because I'm more physically fit because I have a genetic difference that gives me an advantage. And that's basically what drives evolution. But so, giving us various combinations. So these are, this is the why introns, are good. May regulate gene expression. These are those switches that I keep talking about. Okay. These are those switches that I keep talking about. They may help with crossing over or encourage crossing over during meiosis to get more genetic variation. But again, this is just, something that we've just started looking over the last maybe 10, 15 years. So when it comes to genetic work, it takes a hell of a long time. And I think I have some videos in the biotechnology one to some of the websites for the Human Genome Project and whatnot. So it's pretty interesting. How do they figure out what these switches are or what these potential switches are? Again, I think I mentioned this last time. They will tag it with bioluminescence. Okay. We've been able to figure it out. We've taken the bioluminescence out of the jellyfish and we've been able to insert it into the genome. And when that protein gets expressed, it glows. So we can figure out what these genes are potentially doing by just tagging them with this bioluminescence. You put the bioluminescence right in front of the gene segment that you want to figure out what it does. And then you can see what it actually codes for, what it actually expresses. And, does it express body plan or scales or some of the fibers within the various different cells and creatures? So it's pretty fascinating. And that's really all this says. I don't really know if there's a short answer question here or even a multiple choice question, to be honest. It's just there to show you, man, that's how they do it. That's how they figure this stuff out. And it's painstakingly. And if I can show that video, if I can figure out how to get that video, you can see how that would be just a boring job to read through all that genetic information to try to figure out how this one varies with this one. Oy vey, you think your life stinks. That would stink. But those people love it. I don't know. I can't figure those people out.